In this lecture, I'm going to continue talking about my working model of evolution and talk in general about how mutation and selection cause evolution, cause change in the frequency of alleles, the frequency of traits in a population over generations. Mutation is a change in DNA sequence, a change in genetic information. That can result in a change in the traits of a body, the traits of an individual. Selection does not act directly on alleles, but acts on an individual. It acts on the individual's phenotype, the individual's traits. So phenotype is where mutation meets selection. So I'll start this story by talking about our Corvallis slugs, a population that has no spots, and then over generations evolving so that there now is a population where there are numerous slugs with spots, so frequency of 0.5. Okay, so then we're going to focus in on this population of slugs as it's evolving over generations. So we're focusing in at this point in the evolution where there still are slugs with no spots. And then we're going to look over several generations here and follow kind of the genealogy, the genetic connection over time between one of these individuals that has no spots and an individual later that does have spots. So we're going to imagine this individual here, a male, mating with a female slug. And so after mating, here's the fertilized egg. Here's the chromosome that came from the male and the chromosome that came from the female. And then this fertilized egg is going to grow and divide, and build a slug body. So here's that slug individual after developmental time has passed. This male slug, within its body, within its testes, there are spermatogonium that undergo meiosis. And that meiosis results in a recombination between the two chromosomes. So these are the homologs here. I've simplified this picture. I'm not showing the sister chromatids. And so recombination is occurring between the homologs. That recombination results in new combinations of alleles across the genetic loci of the chromosome. And there are many cells undergoing meiosis. There's a population of sperm, so that results in a lot of new haplotypes. So a lot of genetic variation is generated. Of course, most of these sperms will never fertilize an egg, so they're just kind of lost. But the sperm that do fertilize an egg are then going to pass that particular haplotype on to the next generation. So here I'm showing the male mating with a female, and I've drawn the female's chromosome green, so indicating that it's very distinct from the male's chromosome. And so I'm imagining that the female here came from a subpopulation that had been not interbreeding with this subpopulation here for a long time. So they've been diverging genetically from each other. And then this kind of rare event has happened where this female slug has ended up in this subpopulation here. Maybe that female, when it was just an egg itself, got stuck on the foot of a bird, and that bird flew across the valley, and that egg ended up here in Corvallis, and grew into this female slug, and then that female slug mated with a local Corvallis slug. So when you have this kind of mating happening between two individuals from kind of divergent subpopulations, that often results in what's called a hybrid, which across the genetic loci, so I'm just imagining 1,000 genetic loci across this chromosome, it results in at all those genetic loci, or at least a lot of those genetic loci, them being heterozygous in the diploid. So maybe 100% heterozygous or 90% heterozygous, something like that. That would be in contrast to an individual mating with another individual within its subpopulation, or maybe even inbreeding, in that individual is mating with another individual that is actually genealogically related, genetically related, like a first or second cousin, something like that. In that case, the resulting diploid, fertilized egg, is going to be potentially like 90% homozygous across the genome and maybe only 10% heterozygous. Okay, so now I'm imagining one of these offspring. I'm just following one of the offspring of this mating. These other lines represent other offspring. And so here is the recombined chromosome after the oogonium of the female undergoes meiosis. So this is the recombinant chromosome after recombination. And then she is mating with a male slug who's up here, who brought in this particular haplotype here. And so this is the individual then that develops from this fertilized egg. Then a female, and so her oogonium undergo meiosis to create this new haplotype that is a result of recombination between these two chromosomes here. And then she's going to go ahead and mate with a male, and I've drawn the chromosome here a distinct color, indicating that this male is coming from a subpopulation that has been genetically isolated for some time from this female's subpopulation. So another kind of gene flow event. And so their mating results in this individual here. And then that male, its spermatogonium undergo meiosis, and here is one recombinant from that meiosis. And then I'm imagining that male mating with a female from within the population, and here's the diploid fertilized egg from that mating. However, here is where I'm imagining that during the germline cell divisions leading up to the spermatogonium prior to meiosis, a mutation has happened. And that mutation is in a gene coding for a protein that is involved in some way with the synthesis or breakdown of pigment. And so one of the offspring from this mating here is the mutant that has spots. So there's a little inconsistency here with a later part in this story. And so when a mutation happens, that's creating a new allele. And because it's just a single allele and I'm showing it changing phenotype, this implies that this allele is dominant. But later you'll see that I actually kind of switch the story and make it recessive. So a little inconsistency, but that's the way stories go, I guess, sometimes. Okay. So here we have kind of zoomed in, looking at the chromosomes, and I've drawn a little red mark where that mutation is. And so mutation plus recombination is the source of genetic variation. And so another thing just to point out about the evolution that's happening here is that there's population turnover. So there's individuals that are born, right, each generation. In addition to that, there's also death. So, individuals are dying and so forth. So individuals are transient. So bodies are just transient. In contrast, genetic information is essentially what travels or exists over evolutionary time. And what kind of travels is not a chromosome itself in a sense, but it's haplotype blocks. And so haplotype blocks arise because recombination does not happen equally anywhere across the chromosome, but there are recombination hotspots where recombination has a higher frequency of occurring or a higher probability of occurring. That results in kind of chunks of DNA kind of traveling through time as a unit, and these are called haplotype blocks. And so this haplotype block here, if we are to kind of follow its history, so going back in time this way, so there's the haplotype block, and then we're kind of following that back this way. There it is again, and then we're going this way over here, and there it is again. So this haplotype block here that we're following actually came in on this lineage over here. In contrast, if we were to follow this haplotype block here, we're kind of following it back, and we're following it back to here. And oops, sorry about that. Where do we go? This way. Sorry. Okay, so yeah, we're following the green haplotype block. We're following that back through its history and back to here. And so it came in over here. So that haplotype block came in on this lineage here. So each haplotype block actually has its own history. And that's actually what these businesses that will infer your ancestry are actually tracking are these haplotype blocks and kind of determining um, the history, ancestry of the different haplotype blocks that makes up a person's genome, and then letting them know they're like 20% of their DNA comes from some part in Europe and 40% some part in Africa and another 40 60 what was that 40 60 another 40% say um, from Central America or something. So something like that. So that's kind of what these genealogy, ancestry, inferring businesses are doing. They're actually kind of tracking the history, ancestry of the different haplotype blocks that makes up someone's genome. Okay, so this mutation has happened and has created a new allele, and we'll call that the P allele. And it has a genetic association with a trait. It's resulting in pigmented spots. So it's resulted in this new trait or phenotype in the population. And that's where mutation meets selection, or the ecology, the things in the environment that impose on an individual's survival and ability to acquire resources. And so selection is acting on the phenotype, the pigmented spots. So maybe the pigmented spotted slug is blending in with its environment better and is getting eaten by birds at a lower frequency than non-spotted slugs. All right, so that results in a differential in survival and therefore reproductive success. And that brings us back here. And now we're just going to follow the evolution here. That results in the change of allele frequency. And so that might involve some selection, maybe some genetic drift influenced the change in the allele frequency of the the P allele. Maybe some gene flow was involved, kind of decreasing the frequency. So over this kind of evolution, at times maybe the frequency of this spotted allele is increasing, at other times maybe it was decreasing, and now we're at this point where it's at a frequency of 0.5. Okay, so to understand mutation a little bit better and how it has a genetic association with traits, we just want to kind of describe a general model of an individual. And so I kind of break that down into several simple questions. So one, what is it? So what is a bird or an elephant? Or what is a sequoia or a lupin? And in general, the answer to that involves kind of a description of its organization, its parts, and the connections between parts. And that's all about talking about its phenotype, its traits. And so that there's kind of a hierarchy of organization. The outward appearance is what we call the form morphology, and that might be on the scale in linear terms of like 1 meter, or you know 0.1 meter. It just depends on what plant or animal or fungus you're talking about. And then of course there's area and volume to think about. Then if you kind of cut that plant or whatever animal and you look inside, then you're looking at the anatomy, and you may be looking at organs or organ systems or tissues, and then you're starting to kind of zoom in to more details of the organization. So you're down more at a linear scale of 1 millimeter, and then you see using a microscope that the tissues are made up of cells, and so then you're down somewhere around 10 micrometers, 100 micrometers. And it's these cells that I think are a really good place to kind of make that bridge between macroscopic and microscopic, between kind of traits and genetic information. And so cells do you can break down cell behaviors into several basic behaviors. They grow, they divide to make more cells. So one cell becomes two, two becomes four, four becomes eight, eight becomes 16, and so forth. The cells differentiate, so they take on different functional organizations, different cell types. So you have some cells that are neurons, some cells that are muscle cells, some cells that are endocrine secretory cells that are secreting hormones into the blood. You have just a variety of cell types, and that's a process called differentiation. In animals, cells migrate. In plants, very important to the growth of the whole body is cell expansions. Cells make all kinds of secretions. In vertebrates, cell secretions are what build bones. And then, very importantly, the cells are constantly talking to each other, so there's signaling going on. And that signaling can be local signals, cells in the same tissue organ talking to each other, or those cells can be systemic, being distributed throughout the body in the blood system of an animal, or in plants within the xylem or the phloem. So that's often kind of hormones. Or in the case of animals, signals could be transmitted by electrical pulses along neurons. And all that signaling leads to the cells coordinating their behaviors so that tissues and organs act as a functional unit, and those tissues and organs are coordinating their behaviors so that the whole body acts as an individual. Okay, so the other question besides what is it, what does it do? So kind of the dynamic nature of the organization. Well, the first thing it does is it builds a body through development, but then it has to maintain the body and repair the body. And then the body carries out behaviors or physiology like an animal, like muscle contractions, thoughts, feelings, digestion, reproduction. Okay, so then the third question is a really important question, and that's how does it do it? So how does a body take energy, materials, and external and internal information and direct those materials and energy into building the body and building that kind of functional organization? And so to get at the answer, we need to go a little deeper into the organization of an individual. And so we got to go down to a lower level of this hierarchy and go inside cells. So cells have compartments, often referred to as membrane-bound organelles, and then their compartments consist very much of macromolecules, polysaccharides. That's often actually uh, a lot of the material that is outside the cell, so kind of the outside compartment. And then there's lipids that make up the membranes, and then there's the proteins. And then those proteins are coded for by DNA, one type of nucleic acid. So those are the four macromolecules. And then, of course, there's also small organics, metabolites, and the subunits that are used to make these macromolecules. And then there's inorganics, water. And then you get down to like protons and electrons, of course, making up atoms. But, you know, down at this level, these simple things, protons and electrons, that's where life really gets started, right? So that's kind of the energy conservation and chemistry, the proton motive force, you might recall, and redox electron flow. So the whole mitochondria powerhouse of the eukaryotic cell is about electron transport chain, so electrons flowing like water and coupling that flow to generating the proton gradient, and then that proton gradient being used to generate ATP, and then lots of different proteins in the cell using that ATP as an input of energy to do the work that those proteins do. And it's those proteins that are really the mechanisms. So when there's a how question, how does something do something? To me, that I think of this word mechanism. What is the mechanism that directs the flow of energy, materials, and information into these processes? And that flow is happening down here in the cells. That's where yeah, okay, that's where it's going on. And so these proteins, the proteins are what are Okay, I'm going to get to that in a sec. The proteins are the mechanisms down here at this cell level. The proteins are directing the flow of energy, materials, and information. And of course, it's proteins that genetic information codes for. DNA codes for proteins. Okay, so all this that we've talked about, the form, morphology, anatomy, everything that makes that up, organization, the cells, the macromolecules, polysaccharides, lipids, proteins, small, yeah, not this stuff down here. So proteins, lipids, polysaccharides, all this up here above this point, that that is part of the body's phenotype. It's part of its traits. So you can talk about phenotype in terms of molecular phenotypes or molecular traits. Cells have behaviors. Um, it's the DNA alone, that is the genotype component here. That's where the genes are and the alleles. And from this genetic information come all kind of phenotypes and traits, in a way, starting with the proteins, and that's what genetic information codes for. Okay, so right, so proteins. So how do proteins account for cell behaviors? Well, proteins, there's diversity of proteins doing all kinds of specific things, but there's really three basic things that proteins do. One is selective binding. A second is catalysis, catalyzing chemical reactions, and a third is allostery. So I'll go ahead and give an example in just a sec, but I just want to come back over here and have us recognize that this hierarchy continues up. So we're talking about the organization of an individual, but individuals make up populations, and populations of different species make up a community, and that leads up to ecology when you incorporate not just the living component of the environment, but also the abiotic, the non-living components. And so this is kind of at the top of the hierarchy. And then down here, this is the bottom, right? So that's the genetic information, DNA. That's where mutation happens. So mutation is kind of a bottom up. It causes change that ripples through the system from the bottom up. And then ecology through selection is then kind of sorting that change that is presented to it in a sense. Okay, so proteins. So selective binding. So in a cell, a protein is synthesized. Remember that a protein is a chain of amino acids that then folds back on itself to form a three-dimensional shape or conformation that has a really cool detailed surface of bumps and grooves and distribution of charge. And now think of that one protein, and you're that one protein in a cell bouncing around the cell. From that protein's perspective, there are thousands of different surfaces that it's bouncing into. But among those thousands of different surfaces, there's one or two that are going to have a surface that match so that they physically can fit together. And when they bump into each other and kind of cozy in close to each other and form that interface, if in addition to fitting together, the charge distribution is positive charge across from negative or no charge across from no charge, then non-covalent bonds form. So this matching of shape is called complementarity. And I just want to remind you here that I mentioned complementarity in the previous lecture, but at a higher level, up when we were talking about ecology and individuals and adaptations, and how certain traits of individuals actually seem to kind of match their ecological niche. So I just want you to keep that in mind for a later conversation when I'm talking about the origin of life. Okay, so selective binding. So that's kind of the first thing. And then after two surfaces bind together, some proteins are able to then change shape after binding. So here's this protein, and we're going to call this the P protein because we're going to relate it back to spots, the trait of spots. And so up here, we see this surface of the protein here is drawn to look smooth, but after binding, and these red lines represent the non-covalent bonds that hold those two surfaces together, there's a change in the shape of this protein's surface over here. And so that's called allostery and coupling because it's going to couple this first event of binding to a second event. The cool thing about non-covalent bonds is that each non-covalent bond is actually kind of weak, a lot weaker than a covalent bond. And so depending on how much surface area is here, there can be a different number of non-covalent bonds. So depending on the partners involved, they can stay together for just a very brief time if there's only a few non-covalent bonds, or they stay together for a lot longer if there are more non-covalent bonds. So it results in a range of dynamic connections between parts. Okay, so the third thing then is catalyzing chemical reactions. So we have this change in shape, and so the binding here is now being coupled to this P protein binding to a substrate, which actually is a pigment. It absorbs certain wavelengths of light, resulting in color color. And so it has complementarity, and it's binding to the surface of this protein, and then this protein is catalyzing a chemical reaction that's resulting in a breakdown product that no longer absorbs light. So it results in no pigmentation, no spots. So the three basic things proteins do: selective binding, allostery, and catalyze chemical reactions. Okay, so this is this step here is selective binding, but also these other steps. But we're going to focus here is where mutation creates change in a sense. So mutation is a change in the DNA sequence that can change an amino acid. And in changing an amino acid of a protein, it can change the surface details of that protein so that that protein no longer has complementarity. It can no longer bind to the partner it normally binds with. And if it does not bind, there's no coupling, there's no catalysis, there's no breakdown of the pigment, so pigment accumulates, in this case, creating spots. So this new allele I'm going to draw as a lowercase p, a small p, because it would be a recessive mutation. Again, this is the part that's inconsistent when I was telling the story earlier and I created it as a dominant, but this actually would be recessive because this loss of function that results in no pigment, if the individual is a heterozygote and it has one functional copy, it's still going to be able to break down that pigment, maybe at a slower rate. So maybe there would be a faint spot, but there's still a functional, half the protein that is expressed is functional. Okay, so that actually brings us into just reminding ourselves of Mendelian genetics. If we're having a heterozygote mating with a heterozygote, then our expected genotype of the offspring would be 1, 2, 1, because this is a situation of dominance and recessive. Phenotype would be 3 to 1. Okay, and then this also just is as a place we can bring into our model of evolution where population genetics fits. So population genetics, you might recall, starts with this foundation of the Hardy-Weinberg equation, which is derived from Mendelian segregation, p squared plus 2pq plus q squared equals 1. The thing is, with population genetics, instead of just thinking of two individuals mating, you're actually thinking of all the eggs and sperm in the entire population, and that represents the gene pool. So you're accounting for all alleles at a particular genetic loci in a population. So we'll come back to that later. Okay, so one type of mutation is a mutation that happens in the coding region of a gene that changes an amino acid and can alter the binding of that protein to its partner. Another very important mutation that will come up in this course that is important to evolution are mutations that happen in regulatory DNA. So here we have the P allele of the P gene, and the coding region is over here, which would be downstream of the promoter. The promoter is where the RNA polymerase is positioned. The RNA polymerase is the enzyme that will make the RNA copies of the gene, and then those RNA copies are translated into protein. So DNA transcription leads to RNA, RNA translation leads to proteins. That's what gene expression is. In addition, there is regulatory DNA sequence, and that's where transcription regulators bind. Proteins called transcription regulators, and those transcription regulators help position the RNA polymerase at the promoter. So you can have a mutation in the coding region, which changes an amino acid and then changes the behavior of that protein. But there can also be a mutation in the regulatory DNA sequence. And so, as far as regulating a gene, that involves incoming signals, so essentially information, that ultimately is in the form of what are called signal transduction pathways and these transcription regulators, and that can turn on expression or it could turn off expression of a gene. But if you have a mutation, just a GC to an AT base pair, that changes the surface of the DNA right at that regulatory DNA sequence so that a transcription regulator cannot bind. Then that gene's expression may be turned off. And so that's another type of mutation. And if that protein is not present, and it's homozygote recessive, so there's no functional protein, then again that could lead to spots, pigment. And so the mutation happens down at this DNA level, and then it ripples up in a sense to altering the traits or phenotype of the individual. And then that's where selection comes in. Okay, so selection landscape refers to the conditions of life. So it's all the things in the abiotic and biotic environment that impose on an individual. And so here we have, our individual, we have the chromosomes, and then the dots represent different genetic loci, and then through expression, I'm just indicating that they're associated with proteins that are involved in processes that account for certain cell behaviors. So in the case of the pigment, that protein was involved, in a sense, in the cell's metabolism, the flow of chemical compounds, chemical reactions, and so forth. Other genetic loci code for proteins involved in cell division or cell signaling or cell secretions and so forth. So this line out here represents the cell behaviors that are being coordinated, and in an association between those behaviors and specific genes. And then those coordinated cell behaviors through development and physiology leads to the traits and phenotypes of the body. And then these genetic loci, through directing the proteins directing cell behaviors, are associated with higher level states of organization that are the traits, the morphology, the anatomy, the behavior of the body. Okay, so that's the individual. And so then we have aspects of the environment that are abiotic, so non-living, so kind of the substrate and soil, the physical properties there, also the nutrients and mineral organics that are available, whether there's shelter in the environment, water availability, temperature, radiation, sun, oxygen, carbon dioxide, and all these kind of aspects of the environment are, in a sense, imposing on the individual's phenotypes and traits, imposing on its survival, maybe causing stress, and on the abilities, the individual's ability to acquire resources that it can then allocate to building and maintaining and repairing the body. So this, again, is where mutation kind of meets selection. And then we have the biotic environment, so other living things in the environment, such as herbivores, predator-prey type relationships, pathogens, parasites, mutualisms, competition between species. That's also imposing on an individual's survival and its ability to access resources. So this is where mutation meets selection. Selection acts on traits, phenotypes, which are associated with one or more genetic loci. So for example, this predator is somehow acting on the individual here on some trait. So yeah, again, if we have a bird and it's eating slugs, it's kind of imposing on the survival of those slugs. And then we have these slugs that have spots that are blending in with the environment. And those spots are in part determined not just by a single genetic locus. We kind of were following one particular genetic locus, that P gene, but potentially the pattern of spots. Maybe the pattern of spots is influenced by another genetic locus. So that could potentially result in a quantitative trait where there are multiple genetic loci influencing the expression of that trait. So often where you just have one genetic locus involved, that often is associated with a qualitative trait. So just bringing this up because in genetics, you probably talked about qualitative traits. And then where multiple genetic loci are associated with a trait, that often associates with a quantitative trait. Okay, so it's not just interactions with the abiotic environment and other species in the community, but an individual is also interacting with other individuals within its population, other individuals of the same species. And that interaction can be a competitive interaction. And so the individual then is in competition and it's allocating resources to building the body, maintaining and repairing. But then whatever resources it has, left, it's going to allocate to producing offspring. And so that's that individual's fitness. Well, the other individuals are also allocating what resources they can to their own fitness. And then that's where the differential comes in. Differential due to a particular trait, spots, no spots. So this kind of competition between individuals and survival and acquiring resources and allocating resources to reproduction is often what falls under natural selection. But an individual is not only competitively interacting with other individuals, but an individual is also interacting with individuals with respect to mating. And so traits that are involved in gaining access to a mate and potentially being choosy about a mate fall under sexual selection. In addition, there are many examples of cooperation. A lot of animals have social intelligence and they are cooperating with each other. And traits that are involved in that cooperation, that often falls under what's called kin or group selection, group being a type of multi-level selection. Okay, so the first two lectures really laying down a lot of stuff, but trying to lay down a working model of evolution, trying to touch on many of the aspects, but then through the rest of the course, we're now going to focus in on some of these different mechanisms and concepts in more detail. All right, well, enjoy the day.